Hi everyone. Today we are going to discuss AFB staining. Zeal-Nielsen staining technique is a differential staining technique. It was initially developed by Zeal and modified later by Nielsen, hence the name Zeal-Nielsen staining. Nielsen used carbon fusion with heat and added a decolorizing agent, acid alcohol, and counterstain methylene blue. The use of acid alcohol in the technique, so it is called acid-fast staining. Now, where AFB staining is used, microorganisms that are not easily stained by basic stains, such as gram staining, native staining, example Mycobacterium, actinomycetes, Nocardia, Cryptosporidium, etc. The microorganisms which we mentioned in the previous slides have thick cell wall made up of lipoidal complexes known as mycolic acid. Mycolic acid on its cell wall making it waxy, hydrophobic and impermeable. Mycolic acids are beta hydroxycarboxylic acid made up of 90 carbon atoms defines acid fastness of the bacteria. Here comes the principle. The Zeal-Nielsen stain uses basic fusion and phenol compounds to stain the cell wall of the Mycobacterium species. Basic fusion in combination with phenol penetrates the cell wall and stains the bacilli bright red. Once stained, they resist decolorization with strong acid. Mycobacterium does not bind readily to simple stains. Use of heat along with carbon fusion and phenol allows the penetration through the bacterial cell. The basic dye in combination with mineral acid forms a yellowish brown compound which easily comes out of the non-acid fast bacilli after decolorization but not out of the acid fast bacilli. Counterstaining with methylene blue is done to form a contrast to red colored acid fast bacilli. Reagents we need Zeal-Nielsen carbon fusion, which is made up of basic fusion, phenol, absolute alcohol and distilled water. Sulfuric acid 20%, the percentage may vary that we can discuss in the later slides. Lafleur's methylene blue. Requirements, we need slides, split lamp, nichrome blue or stick, glass marker, normal saline, distilled water, liquid paraffin, gas piece and light microscope. Procedure, take a new slide and make an oval shaped mark at the center by using a glass marker. Make a smear from blood tinged or yellow purulent portion of the sputum using a stick in the pre-marked area. Allow the smear to dry for 15 to 30 minutes. Fix the smear by passing the slide over the flame for 3 to 4 times. Place the slide on the staining glass rods with the smeared side facing upwards. Pour filtered carbon fusion over the slide so as to cover the entire slide. Heat the slide underneath until vapors start rising. Do not allow carbon fusion to boil or the slide to dry. Continue the process for 5 minutes. Wash both sides of the slide with tap water. The next step is to cover the slide with 20% sulfuric acid. The red color of the preparation is changed to yellowish brown. After about a minute in the acid, wash the slide with water and pour on more acid. Repeat this process several times. The object of washing is to remove the compound of acid with stain and allow fresh acid to gain access to the preparation. The decolorization is finished when after washing the film is only faintly pink. Decolorization generally requires contact with sulfuric acid for a total of at least 10 minutes. The final step is counterstaining. Counterstain with Lafleur's methylene blue for 15 to 20 seconds. Wash with water. Place the slide in slide tray and allow it to dry. Put a drop of oil in the stain area and observe under oil immersion 100x magnification. Let's summarize. Step 1. Primary stain by Zeal-Nielsen carbon fusion. Step 2. Modern, that is to fix the stain by heating. Step 3, decolorization by 20% sulfuric acid. Step 4, counter staining by Loeffler's methylene blue. Now discuss about the modifications. Over the years, lot of modifications have been made. The widely practiced cold method is Cabot's methylene blue stain, Canyon's methods, and modifications of sulfuric acid. A solution of 3% hydrochloric acid can be substituted in place of 1% sulfuric acid. The sulfuric acid solution does not decolorize as strongly as the hydrochloric acid. This makes it useful for staining organisms that are weakly acid fast, such as Nocardia. Brilliant green may be substituted for methylene blue as a counterstain, resulting in non-acid fast organisms appearing green rather than blue. Gabbert's methylene blue method is widely practiced method. Gabbert's methylene blue can be prepared with 1 gram of methylene blue, 20 ml of sulfuric acid, 30 ml of 95% ethanol, 50 ml of distilled water. Here comes the cold method, the two-step procedure, Gabbert's methylene blue method. Earlier we discussed 4-step procedure, that is certain staining, that is heat method. Now place the slide on the staining glass rods with the smear side facing upwards. Pour basic fusion phenol over the slide so as to cover the entire slide. Allow to stand at room temperature for 10 minutes. Wash the slide with tap water. Counter stain with Cabot's methylene blue for 2 minutes. Wash with water. Place the slide in slide tray and allow it to dry. Put a drop of oil on the stained area and observe under oil immersion 100x magnification. Now, Kenyon's cold method. The procedure for Kenyon staining is similar to Ziel-Nielsen stain but does not involve heating. The Kenyon staining method uses carbon fusion as a primary stain followed by decolorization with an acid alcohol solution and methylene blue as a counter stain. Kenyon carbon fusion has a greater concentration of phenol and basic fusion and does not require heating in order to stain properly. These are Zedden stain methods for spores, Muller's method, Dorner's method, Schaffer-Fulton stain, Muller-Chermak, Tergit-Hall method. Results can be observed acid-fast bacilli as bright red bacilli against blue background. Other pus cells, squamous epithelial cell, elastic fibers all are stained with blue. Now interpretation. Acid-fast bacteria retain primary dye, that is carbon fusion, as we discussed in the earlier slide, pink in color. Example, Mycobacterium, Cryptosporidium, Nocardia, Isospora, etc. Non-acid-fast bacteria does not retain primary dye, that will be in blue color as we said in the previous slide. Grading of microscopy smears for Mycobacterium tuberculosis. If we see 10 or more AFB in each oil immersion field, that is each oil immersion field, that will be 3 plus. 10 or more AFBs in entire smear, that will be 2 plus. 3 to 9 AFB in the entire smear, that will be 1 plus. If we see only 1 or 2 AFBs in the entire smear, that will be scanty or to repeat the smear. Next is implications. Ziel-Nielsen method of staining is useful in detection of acid-fast organisms. Number of bacilli in the smear may be counted and correlated with infectiousness of the person. This is important for epidemiological purpose. This is useful in following a patient's response to treatment to know the drug resistance if organism fail to decrease after therapy in the smear. The possibility of drug resistance must be considered. How to prevent the false positive sputum results? Ask the patients to rinse the mouth with water before collecting the sputum sample. There should not be any food particle or fiber in the sputum sample. Use only new unscratched slides. Always use filtered carbon fusion. Do not allow carbon fusion to boil during staining. Decolorize adequately with sulfuric acid. Never touch the oil immersion applicator to a slide. Always clean the oil immersion lens after screening each smear. How to prevent false negative sputum results? Make sure that sample contains sputum and not just saliva. There should be enough sputum, at least 2 ml. Select thick purulent portions to make the smear. Fix the smear with correct length of time. Prepare smear with adequate material. Stain with carbol fusion for 5 to 7 minutes. Do not decolorize with sulfuric acid too intensively. Examine every smear for at least 5 minutes, observing at least 100 fields before recording as negative. The discussion part. This is the main goal of this video. What are the questions the examiners can ask you? What do you mean by acid fastness? Acid fast bacteria do not stain readily with dilute solution of dyes, but they do stain when heated with a strong dye solution, and once stained they resist decolorization even by strong mineral acid. Name the various acid fast organisms. Tubercle bacilli, leprobacilli, atypical mycobacteria, Nocardia species, Legionella mygdadae, Rhodococcus species, bacterial spores, oocysts of Cryptosporidium pavum, Isospora belli. What are the factors which affect acid fastness of an organism? Age of colonies, medium on which growth occurs, ultraviolet light, lipid content, and integrity of the cell wall of the organism. Why tubercle bacteria are acid fast? Due to the presence of mycalic acid in their cell wall. Why heating of carbol fusion is necessary in Ziel-Nielsen staining? Heating helps in penetration of dye, that is carbol fusion, through waxy coat surrounding the cell wall of the mycobacteria. What are the other methods of acid fast staining? Kenyon acid fast stain, fluorochrome stain. Why Kenyon acid fast stain method is known as cold method? The Kenyon stain is known as cold method because the high concentration of phenol in the reagent serves to dissolve the lipid material in the cell wall, allowing penetration of carbon fusion dye without the use of the heat. Which method of staining is most useful screening procedure? Fluorescent dye staining. What are the advantages of fluorescent staining over Ziel-Nielsen staining? Fluorescent staining is more sensitive, no heating is required, slide is scanned under low power and hence scanning is quick. Positive fluorescent smear may be restained by Ziel-Nielsen or Kenyon procedure, thereby saving the time needed to make a fresh smear. Mycobacteria appear as bright luminous yellow rods against a dark background. What are the drawbacks of fluorochrome staining? Many rapid growers, example Mycobacterium coloniae, Mycobacterium fortuitium, may not appear fluorescent with these reagents. Fluorescence microscope and reagents are expensive. Expert hands are required to distinguish between positive bacilli and artifacts. What are the limitations of acid fast staining? Smear must always be correlated with culture results. Organisms other than mycobacteria may demonstrate various degrees of acid fastness. Rapidly growing mycobacteria may stain poorly or acid fast variable. What is the number of bacilli which must be present in the sputum for detection by direct microscopy? 50,000 to 1 lakh bacilli per ml. What is the minimum number of bacilli which must be present in the sputum for detection by direct microscopy while using standard concentration technique? 10,000 per ml. Can we use counterstain other than methylene blue? Yes, malachite green may be used. What is the disadvantage of malachite green? This is a strong counterstain and may mask the presence of AFB. Can we decolorize the smear with acids other than sulfuric acid? Yes, nitric acid or hydrochloric acid. What is the role of phenol in carbon fusion? It acts as a mordant. It also makes the cell surface easily penetrable for basic fusion by dissolving fats. Which method is more sensitive, Ziel-Nielsen or Kenyon? Ziel-Nielsen is more sensitive. Weak acid fast stains of rapidly growing species may stain better with Ziel-Nielsen than Kenyon. What is the concentration of sulfuric acid used for decolorization of various acid fast organisms? Microorganisms for tuberculosis 20%, atypical mycobacteria 20%, leprae 5%, cryptosporidium 1 to 5%, nocardia species 0.5%, bacterial spores 0.25 to 0.5%. How can you differentiate Specma bacilli present in urine from Mycobacterium tuberculosis? Mycobacterium tuberculosis is acid and alcohol fast, while Specma bacilli, that is commensals, are only acid fast. These are the exercises. Thank you. In the next video, we will discuss about the gram staining and Q&A about gram staining. Thank you.